Lecture 22. Polymicrobial infections. Dental caries. Periodontal infections. Oral microbiology. Dr. Manish Kumar. Microbial ecology of the mouth. Constant introduction of new microorganisms. Many different environments. Tooth surface, gums, tongue, salivary duct and glands, and more. Complex microbiology. www.microbilibrary.org. Gingivitis. Gums are red and swollen and may bleed easily. Brushing and flossing can help improve this condition, or professional care may be needed. If plaque remains on the teeth, it hardens to form tartar or calculus, which irritates the gums. The tartar or calculus cannot be removed by brushing. Eventually, the gums detach and pull away, recede along the length of the teeth. Tartar on the teeth may need to be removed by having the teeth cleaned before the gingivitis will improve. Periodontal disease. If the gingivitis is not treated or controlled, it can progress to a more serious condition, periodontal disease, where the bone holding the teeth in place is destroyed. Eventually, the gums may pull away from their original position, exposing the roots of the teeth. This allows decay and infection into tooth roots and also into the bone that supports the teeth. As the bone dissolves, the teeth will become loose. The benefits to the pregnant patient are many. If good oral health is promoted, this promotes good systemic health. Anticipatory guidance has the potential to prevent early childhood caries. Gestational diabetes. High blood glucose levels in pregnant women who have never had diabetes. Pregnant women who develop GT are at greater risk for periodontal disease than women who do not. If periodontal disease occurs, control of GDM is more difficult. Preterm births and periodontitis. Severe periodontitis leads to high levels of prostaglandins in the blood. High levels of prostaglandins are associated with early uterine contractions, early birth and low birth weight. Preeclampsia and periodontitis. A vicious cycle. Patients with periodontal disease are twice as likely to develop diabetes. Poor glycemic control is associated with threefold increase of periodontal disease. Periodontal disease worsens glycemic control. Treatment of periodontal disease results in 10–20% improvement in glycemic control. References. Mealy, B.L., Periodontal Disease and Diabetes, A Two-Way Street. J.M. Dendasic 2006. 137, 26–31. Demer, R.T., Jacobs, Drive, Zverev, and Periodontal Disease and Incident Type 2 Diabetes Mellitus, Results from the First National Health and Nutrition Examination Survey and its Epidemiologic Follow-Up Study. Diabetes Care 2008. 31-7, 1373–9. Moore, Pennsylvania, et al. Type 1 Diabetes Mellitus, Xerostomia and Salivary Flow Rates. Oral Surg Oral Med Oral Thal Oral Radial and Dodd 2001. 92, 281–91. Promsut, A., et al. The Effect of Periodontal Therapy on Uncontrolled Type 2 Diabetes Mellitus in Older Subjects. Oral Dees 2005. 11, 293–8. Tooth Structure. Source www.nih.gov. Gingiva. Streptococcus mutans. Dental caries, tooth decay, other Streptococcus also involved. Only found when teeth present. Metabolizes sucrose and starch. Produces lactic acid and glucans, sugar polymers. Adhesion, Kreth et al. 2008. At high cell density becomes acid resistant and competent stimulating peptide, Corn signaling. Streptococcus mutans. 3 key factors. Sucrose converted to dextran, a sticky material which help adhesion. Production of acid for demineralization of tooth. Ability to survive in low pH. The process of tooth decay. S. mutans produces dextran from sucrose, allowing plaque formation in pits and crevices on enamel. Lactobacillus and other bacteria produce acids from sugars via fermentation, which dissolve the enamel. Decay continue into the dentin and pulp. Tooth decay. O slides and notes, script. Periodontal disease. Bacterial colonization and varying degree of inflammation. Acute necrotizing ulcerative gingivitis, a NUG, trench mouth. 1. Necrotizing ulcerative gingivitis, NUG. 2. And periodontitis, NUP. Periodontal infections. Aggregatibacter actinomycetem comitans. Porphyromonas gingivalis. Nerelliversithia. Treponema denticula. Polymicrobial. Tooth loss, other problems, heart, etc. NIH. Tooth microbiology. Conditioning film from saliva. Early colonizers include Streptococcus gordoni, Streptococcus aureus, and Actinomyces neslandi. Streptococcus mutans colonizes shortly thereafter. High shear forces, coaggregation. Microbial interactions are very important. Palmer et al. Infect. Immune. 69, 5794, 2001. The microscopic and microscopic appearance of plaque. A. Disclosing tablets containing vegetable dye stain heavy plaque accumulations at the junction of the tooth and gingiva. B. Scanning electron micrograph of the plaque biofilm with long filamentous forms and corn cobs that are mixed bacterial aggregates. Representative confocal micrographs of 4H and 18H2 species biofilms grown in flow cells. The primary immunofluorescence indicates S. aureus, blue, A. neslandi, green, and F. nucleatum, red, and shows that there is intimate contact among species. Perius et al. Apple. Environ. Microbiome. 2009. 75, 3250 to 3257. Polymicrobial interactions with A. All dental organisms live in mixed microbial environment. Examples of polymicrobial interactions with A and S. gordoni. A. Nutrition, Brown and Whiteley, 2007. A. Immune resistance, Ramsey et al. 2009. Shows how A can grow in mixed environment and also survive immune system endocarditis. Glucose, fructose, and L-lactate consumption by A. actinomycetem comitans. A. actinomycetem comitans was grown in the presence of equimolar 2 mm glucose, fructose, and L-lactate, and the disappearance of these carbon substrates was evaluated over time. Experiments were performed in duplicate. A. actinomycetem comitans cauda and opia are induced on H2O2 exposure. A custom Affymetrix genetics was used to examine gene expression of A. actinomycetem comitans colony in flow cell biofilms in the presence and absence of 1 mm exogenous H2O2. Fold changes were determined from 4 pairwise comparisons and determined to be statistically different for cauda and opia p less than 0.05 using genetic operating software version 1.4. B. RT-PCR was used to verify cauda and opia induction in colony biofilms on following exposure to H2O2. The constitutively expressed gene CLPX was used to standardize GNR template levels, and planktonic-grown bacteria were used to assess the impact of enhanced duration on basal transcript levels. Co-culture with S. gordoni enhances A. actinomycetem comitans resistance to killing by human serum. Fold increase in survival of A. actinomycetem comitans on exposure to 50% vol/vol normal human serum when grown in monoculture. Co-culture with S. gordoni plus heat inactivated catalase A plus Sg, and co-culture with S. gordoni plus catalase A plus Sg plus cat. Ratios were calculated as follows. Colony forming units present after serum treatment / colony forming units present after heat inactivated serum treatment. Error bars represent some. It is important to note that cell numbers were similar with and without catalase in the heat inactivated complement cultures. Asterisk p less than 0.01, asterisk p less than 0.05 via student's t-test, n equals 3. Model for the role of H2O2 as a mediator of A. actinomycetem comitans. Resistance to innate immunity. Enhanced levels of H2O2 produced by S. gordoni during plaque growth stimulate inflammation, leading to an influx of innate immune modulators, including complement and neutrophiles. A. actinomycetem comitans responds to rising H2O2 by induction of cauda and opia, which, in turn, enhance resistance to innate immune effectors. On. Recruitment to the site of inflammation, neutrophiles increase the levels of H2O2 and further stimulate induction of cauda In contrast to the often predictable nature of laboratory culture growth, an organism such as Actinomycetum comitans likely adopts different physiological profiles as it moves from one tissue environment to another. In spite of the many potential nutrients present in host tissues, pathogen physiology during infection remains a largely unexplored field. The RNA-seq approach used provides a mechanism whereby pathogen physiology can be explored during infections. Other dental pathogens. Porphyromonas gingivalis periodontal disease. Some oral pathogens have been associated with heart disease, Actinomycetum comitans, P. gingivalis, S. mutans, other Streptococcus, E. canella, and possibly other organs. Dental professional diagnose caries based on the tooth condition. Cultures are not routinely performed. Brushing and flossing to remove plaque. Fluoride treatment. Scaling/deep cleaning, removal of plaque. Debridement, removal of dead tissue. Broad-spectrum antibiotics given if necessary to treat infection. Treatment and prevention.